Western blotting. So we went from the north, now we're going to the west. Western blotting is similar in its process to southern and northern blotting, which are used for DNA and RNA respectively. However, Western blotting is used to quantify the amounts of target protein in a sample. Western blot is considered a semi-quantitative because it provides a relative comparison of levels, but not an absolute measure of quantity. So I would group this in the same manner that I would say that um, a northern blot is quantitative. You can use images and analyze those images to determine whether or not you actually have like you can get a quantitative measure of a product. Why is this technique done? It's for the detection and identification of specific proteins in a sample. So think about it as, if you think about the central dogma of molecular biology of DNA, RNA, protein, this tells you if your northern blot tells you if that RNA transcript is present, and then your western blot tells you if that protein product is generated. It's commonly used in cancer research to identify proteins that are linked to tumors. So, if some doing Western blot analyses of thousands of people with and without tumors, then you'd be able to say, oh, well, you'd be able to draw correlations between presence of a protein product and whether or not someone has a particular type of cancer. Now, what this process consists of is it's very similar to northern blotting. Um, it's very similar to northern blotting because what we're doing is we're going to first run a gel electrophoresis. Now, this is not an agarose gel. This is instead something like polyacrylamide gel. You're going to run that gel, then you're going to transfer the proteins using an electric current onto a nitrocellulose membrane. Then you're going to incubate that nitrocellulose membrane with an antibody, because that antibody is going to be very, very selective for your protein. Then, and this is something that's similar to northern blotting because of the fact that with northern blotting, because of the fact that you're going to incubate that membrane with a probe, then you're going to expose that membrane to either to a autoradiograph, and you're going to detect whether or not that protein is present or whether a protein is present. Now that antibody could be a radioactive antibody and it has a radioactive label to it, or more commonly there's a fluorescent label on that antibody, so you don't have to use radioactivity to tell whether or not a protein is present on a membrane. Okay, now that experimental process consists of first and foremost detection. So after you've actually run your gel, then transfer those proteins from your gel onto your membrane, you have to use something known as a blocking agent. What this is going to do is prevent non-specific binding. So just as we said, an antibody is very specific, but you still do have to do something to prevent kind of non-specific binding. And what you're going to use is, well, you can either use a purified protein, and what some people use in general is BSA, so that bovine serum albumin. Bovine serum albumin, we used it to develop a standard curve in our lab. Bovine serum albumin is used time and time again for any number of other techniques. It's just like a widely available protein. It's cheap, it's easy to work with, and so you can use lots of standardization techniques for that bovine serum albumin. Alternatively, what people will use as a blocking agent is milk. They'll use 2% cow's milk or whole fat cow's milk, and that will serve as your blocking agent. So all of those milk proteins will um, bind to proteins that have been transferred onto your nitrocellulose membrane. Okay, so after you've done that blocking step, you're going to incubate that nitrocellulose membrane with your primary antibody. So that first antibody, that primary antibody, is going to detect your protein of interest. Then what you're going to do is rinse that sample, or rinse that membrane. So let me elaborate here. Rinse membrane. Basically to get rid of excess proteins that are in that solution, rather than proteins that are bound to that nitrocellulose membrane. Then you're going to incubate that with a secondary antibody, and that secondary antibody, what's it going to do? It's going to detect your primary antibody. And then again, after you've done that incubation, you're going to rinse the membrane again. So basically, if you, this line right here, or actually I'm going to do, let's see. Okay, so, oopsies. So this flat line is my membrane. This red bubble there, that is the protein, that is my protein that is immobilized on my nitrocellulose membrane. Here is my primary antibody that has bound to my protein of interest, and then here is my secondary antibody that has bound to and found my primary antibody. And my secondary, so primary, my secondary antibody has something like a bioluminescent signal on it, and that will be produced whether or not my protein is there. So basically what I'm looking for is, or what I'm doing is kind of, I'm seeing, my result is going to show me whether, or is going to show me a bioluminescent or a X-ray signal, and that's going to tell me that my secondary antibody is there. Okay, my secondary antibody is going to tell me if my primary antibody is there. My primary antibody is going to tell me if my original protein is there. So that's kind of where all of this sort of bounces around and lands. Now, X-ray film is commonly used to analyze the results of a Western blot, but you do not have to do that, because if you're using a bioluminescent signal, you're not using radioactivity, so you don't have to use that X-ray film. Instead, you would use an instrument known as a chemidoc, which is a gel imaging system, or instrument. Change that, instrument. Qualitative analysis can be done by comparing sizes of bands as well, and more specifically, quantitative analysis can be done through molecular weight comparisons. In addition to that, what you're able to do is you are ultimately able to look at kind of the density of those individual bands, and so that's why this is a semi-quantitative technique and method. Okay, excuse me. So mostly for detection of proteins, is developed in the late 70s, can be used for detection of antibodies to viruses. It's found autoimmune disease and allergies in clinical practice, and can be quantitative or semi-quantitative based on the types of reagents that are used. This is definitely a technique that is used for an antibody test. So if somebody's had a virus for COVID-19, for instance, whenever people are talking about the so-called antibody test, what they're talking about is a Western blot. Whenever somebody's talking about another the alternative test that people seem to be talking about quite a bit is the real-time PCR test. So that's going to be looking for the presence of the active virus, whereas this Western blotting technique would be looking for, has somebody had the virus because they have antibodies that have already been developed against this virus. Alternatively, if the virus was in high enough concentration, you could maybe detect specifically a protein from that virus. Okay. The impact of Western blotting technique can be very, very valuable to scientists and health professions in detection of specific antibodies. Gives us insights into factors about a target protein. It can be a challenging technique because of the instruments that are involved. But this is what is being used to, one, detect whether or not somebody has COVID-19, but also it is being used all the time in development of a vaccine, and or treatment.